talking about the reproductive system. Reproductive physiology. Title of this lecture is the lecture where we are going to build up to the point at the end where we talk about the making of babies. We'll talk just briefly about the developmental stages of a baby, going from like what you see there on the left at 4 weeks to the point where the baby's at full term. They're at 36 weeks. As you can see, obviously there's a lot of growth and development, especially in the early stages. We'll get into that just briefly, but for the most part, that'll just be a little bit at the end. Let's first introduce reproductive cell division, which is the process in which we produce our gametes, which are the cells that we need to make babies with. So we're talking about sex cells, which are called sperm in men, eggs or ova in women. Have the fancier term known as gametes. And when those unite through the process of fertilization, they fuse together, forming a zygote or fertilized egg, a single cell that we all begin our life as. Okay. The sperm that we'll talk about are produced in the male reproductive organs known as the testes, eggs produced in the female reproductive organs known as the ovaries. We've talked about those pairs of organs before. In our endocrine lecture, we learned that the testes produce testosterone. In that same lecture, we learned about the hormones of the ovaries like estrogens and progesterone. So we'll talk more about those again today and into Thursday as we kind of move further into the lecture. If you look at this illustration, it's a pretty simplistic view of the life cycle found in humans, where as adults, multicellular diploid organisms, we go through a process called meiosis, which produces the sperm and the eggs, which are haploid gametes that then fertilize and begin a whole new life cycle with a diploid zygote, a single cell that eventually through mitosis and development can start the whole process over with an adult producing eggs and sperm again. Now, we've talked about mitosis before, back when we talked about cells. If you think back to that lecture, we talked about prophase, metaphase, anaphase, telophase, the 4 steps involved, where cells duplicate their material and split in half. And that's what happens with that single zygote or single cell zygote. And that becomes 2 cells, that then becomes 3 cells, or 4 cells, not 3, but 4, and then 8, and then so on and so on and so on. And here we are as adults, made up of trillions upon trillions of cells. And we didn't talk about it at that time though, which is meiosis, and I want to talk about that now. Okay, so reading through somatic cells, which include any cell in the body other than a gamete. That could be a bone cell, a muscle cell, neuron. I mean, it's just anything other than an egg or a sperm. They contain 23 pairs of chromosomes, and a pair is 2. So you take 23 times 2. That gives you a total number of 46 chromosomes. Now, one member of each of those pairs is inherited from each parent. So in other words, we have 23 chromosomes that we got from dad, 23 chromosomes that we got from mom. These 2 chromosomes that make up each pair are called homologous chromosomes or homologs. You'll sometimes hear me call them homologous pairs, because that's what they are. They're in pairs. And we number them 1 through 22, pair 1, pair 2, pair 3. But then the 23rd pair, they don't go by numbers. Instead, they go by X and Y. And females, it's X and an X chromosome. In males, it's an X and a Y. Okay. Now, located on those pairs of chromosomes are similar genes arranged in the same or almost the same order. Okay. If I kind of just explain that by going to this next page, here we can see an example of a homologous pair. They always use blue or a kind of darker shade like this to denote the paternal chromosome. That's what was given to the offspring from dad. And then red's pretty commonly used for maternal. Okay. These chromosomes are not necessarily identical, but they do contain the same genes. And I said they're not necessarily identical, like looking here at these letters. Notice there are two big B's here in the paternal chromosome, but in the maternal, they're lowercase b's. Okay. Now, I'm not going to get into genetics. This is not biology class. But let's say that this is the gene, this segment of the chromosome that codes for the color of your eyes. And the big B's are going to code for brown eyes. But the little b's code for blue eyes. Okay. They're both coding for the same thing, the color of your eyes, the same physical trait. But mom and dad aren't genetically identical, are they? So when they pass off their offspring, or they pass off their genetic material to their offspring, there's a good chance that the paternal chromosomes and the maternal chromosomes aren't going to be genetically identical, at least for each of the different genes. Sometimes they are identical. Sometimes you find BB, lower, uppercase, and then same thing, BB, uppercase here. But sometimes you see it like it is right here where they're not. And we can see it with 3 separate chromosomes in all 3 examples. They're not identical to one another. Okay. Because somatic cells contain 2 sets of chromosomes, we call them diploid cells, symbolized with the abbreviation 2N, which tells you that there's 46 total chromosomes in those cells. Okay. During the process of meiosis, which is a reproductive cell division, occurring only in the gonads, which are the ovaries and testes, the production of gametes occurs where the number of chromosomes is reduced by half, leaving each sex cell with only 23 chromosomes, which is often referred to as the haploid number, abbreviated with just the letter N instead of 2N, telling you that there's only 23 chromosomes. And to get to this point, we have to perform 2 successful stages of meiosis, meiosis 1 and meiosis 2. Each of those stages has 4 phases: prophase, metaphase, anaphase, telophase. So typically what you're going to see is prophase 1, metaphase 1, anaphase 1, telophase 1, which goes from 1 cell to 2 cells. And then those 2 cells split, and that's what we call prophase 2, metaphase 2, anaphase 2, telophase 2. So let's talk about those here on this page. Okay. Meiosis 1 is a division that begins with our diploid stem cell. Okay. And as a result of this process, we end up with 2 cells, both of which have haploid number of chromosomes. So this is where we go from 46 chromosomes to now each only having 23. Okay. During this process, the homologous chromosomes separate. So those pairs that we just talked about are separating. And so why we call this, or why we see this number going from diploid to haploid, is because now we don't have the pairs. They've split. So this has 23 chromosomes. This has 23 chromosomes. Okay. During meiosis 2, those 2 haploid cells split, which results in, from our original 1 cell, the formation of 4. And we still see them being haploid because we did not divide the number of chromosomes, but instead we separated what we call the sister chromatids. Sister chromatids are like the 2 halves of a single chromosome. Okay. We kind of just look at this blue one here. They're made up of 2 sister chromatids. It doesn't look like there's 2 separate things, But instead of thinking this like the letter X, think of it like two sideways V's. Okay? And they're just connected right here at its centromere. Okay? This is what will split during meiosis II. Okay? Now, during meiosis I, once again, we separate homologous chromosomes, but something special happens, and that's what this figure actually up here in the top right is showing you. What we call crossing over occurs. Whereas we get those chromosomes ready to split, those homologous pairs, those paternal chromosomes and maternal chromosomes all line up together, and they get really closely associated with one another to the point where they can actually exchange segments, okay? Which is going to be important because that helps increase genetic variability. In other words, it helps produce offspring that are always going to be a little bit genetically unique, unless you're making identical twins, which is where the DNA is actually the same. That's why they look exactly the same, which is exactly what my dad is. And I have two nephews that are genetically identical twins. Okay. So crossing over is something that happens in prophase I. Basically, if you see the terms homologous chromosomes, and you can see that here and all these different definitions or descriptions of the steps, you know you're dealing with meiosis I. If you see it described as going from diploid to haploid, you know that's meiosis I. Okay, we form two haploid cells at the end, and then if you see two haploid to four haploid, or if you see anything about sister chromatids, you know you got to be dealing with meiosis II. Okay, we're not going from 23 now to 11 and a half, right? Not half of 23. We're still staying at 23 chromosomes, so all four end up with 23 chromosomes. Okay. Now, what this illustration is showing you is actually just two pairs of homologous chromosomes. But remember, we'd have 21 more pairs because there's 23 total. And to also increase genetic variability during metaphase I, when they line up along that metaphase plate, it's not like all the paternals are on one side, all the maternals are on the other. They're going to just kind of randomly align themselves, which means when you end up with those four cells, they're all going to be genetically unique to one another. And so if this is a sperm that develops from this cell, another sperm, another sperm, another sperm, every one of those sperm would be unique. Okay. Now, let's watch a little clip to kind of help make some sense of this stuff. I'll find a short one. Hopefully you remember prophase, metaphase, anaphase, all that stuff from way back at the beginning of the semester. That's okay.
The number one reason is not about sunlight or weather or even politics. It's
the cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs.
Okay, so now we see there was a pair there, there's a pair here, there's a pair there. So they've got 3 pairs. Remember, there'd be 20 more. It's going to be way too hard to show all 23 pairs, so that's why you're not seeing them in this video, nor are you seeing them all in the pictures there on your lecture.
Chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The two homologs trade genes in a process called crossing over. This forms new hybrid chromosomes.
So yeah, I can see they've switched segments.
Place maternal and paternal chromosomes randomly on each side of midline.
So here's our,
another shuffle that can lead to a host of different genetic outcomes.
now we're in metaphase 1. We're down the middle.
Finally, spindle fibers pull the homologs apart.
Anaphase 1, and now we're starting to split the
This leaves the
cells.
sister chromatids intact.
So there's your telophase 1 right there.
With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two.
All right, so now we're starting prophase 2.
The second division produces 4 unique sets of chromosomes.
Metaphase 2, now anaphase
Because they
2.
carry half the genetic information, we call these haploid cells.
And here comes telophase 2, and we're going to end up with 1, 2,
Many of
3,
them
4
will join the
cells.
sperm and eggs, the reproductive cells called gametes.
Does that make sense? All right, let me just draw something up real quick, and hopefully this will make it seem super simple. I'm gonna do it like this. So if we start with our one cell, okay, and we've got a couple pairs, I'll just draw 2 pairs, okay. I'm gonna draw the black cells here in maternal chromosomes and my red representing maternal chromosomes, okay. So here's our cell with the nucleus as it starts, and then as that dissolves, we then do the crossing over and all that wonderful stuff. We end up with 2 cells, okay. Now I'm going to show you what happens when crossing over takes place and why this is important. Okay, let's pretend that the crossing over has taken place with just this pair here, okay. So we're going to see something like this has now happened. Oops, I drew it wrong. Sorry. Let me start it over. I messed it up. I'm trying to draw 2. Let me just start with 1 pair. All right, so we've got 1 pair of chromosomes. I'm not even going to worry about the 2 pair because I'm just going to confuse myself. Right now, we've got a set of chromosomes, a set of pairs, okay? And if they cross over, let's say the crossing over resulted in something like this on that one, okay? And now, what we're left with as a result, this is meiosis 1, okay? These 2 are not unique, or they're not identical, are they? Okay. What we're going to see that's even more important is that during meiosis 2, we're going to end up with, there's one sister chromatid that's broken free. There's the other, but now those 2 aren't going to be identical to one another, nor will they be identical to these 2. And so all 4 of these haploid cells are genetically unique, and that's just showing you 1 pair. Keep in mind, you've got 23 pairs and they can cross over, they can line up uniquely, so we can end up with a whole bunch of different variability at the end, which is great. You want genetic variability. Like I said, this is not biology, it's not genetics, so I'm not going to really get into any more detail than that. But that's a benefit of reproductive cell division, that, you know, if you're talking about like bacteria, they don't reproduce sexually, they do asexually, and they don't have that increased genetic variability like we do. Now, what we're going to do at this point, we're not going to get to do it very much, is start in on the male reproductive system. Get through that and then dive in on the female reproductive system, which will not be accomplished today. We only got 5 minutes. But we're gonna start by talking about the testes, a pair of oval glands, which are the actual site of sperm production in males, where the process of spermatogenesis takes place. Spermatogenesis is the fancy term to describe the formation of sperm. A fancy term for sperm is spermatozoa. Okay, we've talked about this pair of organs before, back when we talked once again about the endocrine system because of the fact that they produce hormones. Okay, but now we're talking more about spermatogenesis, although we will talk about testosterone and how that helps regulate spermatogenesis. Within the testes are a highly coiled set of tubes called the seminiferous tubules, which is where the process of spermatogenesis takes place. The cells called spermatogenic cells are the ones that actually do the sperm formation, which makes sense. Spermatogenic cells perform spermatogenesis. You also have Sertoli or sustentacular cells. They also can be described as nurse cells. I usually just call them Sertoli cells, which are going to help form what's called the blood testis barrier, which prevents your immune system cells from responding against those developing sperm cells. So in other words, it keeps those immune cells we talked about a few weeks ago from attacking and destroying your sperm cells and effectively killing them off. Okay. Then there's the Leydig or interstitial cells located not in the seminiferous tubules, but instead between them. And those are important because they secrete testosterone. Okay. Now, very closely associated with your testes are the scrotum, which is the sac that is going to essentially enclose them, made up of hair and superficial fascia, hanging down from the base of the penis. Okay. Most importantly, the scrotum is external to the body to place, and it's kind of weird how it says external to the body. Obviously, they're not, but they're external to the core part of your body. They keep the testes in an environment that's around 3 degrees Celsius below core body temperature, which is important for proper development and storage of sperm. There's a pair of muscles that help keep this lower temperature. That shouldn't say maintain pressure, that should be maintain temperature. I think I typed that up wrong, but the dartos muscle, which causes wrinkles to form in the scrotum, which thickens the wall of the scrotum to reduce its heat loss when the temperatures are cold, and the cremaster muscle, which can contract and lift the scrotum up closer to the body when those external temperatures are cold. So when it is cold, the scrotum comes up closer to the body and it thickens up because of these muscles, which keeps those testes warm. And when it's hot, they do the opposite. They both relax. And so the testes are farther from the body and the scrotum is much thinner, so the testes stay much cooler. All right, we're going to stop right here. We're going to start fresh with this process of spermatogenesis on Thursday and get into it. I can't get through everything that's discussed here in the next couple slides in 2 minutes. So we'll stop here and start fresh on Thursday.